Now we're going to analyze variants using JupyterLite. I already pre-created, if you will, notebook here. So one thing you need to make sure is that this window manager is on. So you need to click it, make sure the checkbox is here. And then we go to visualize, select JupyterLite, and here is the notebook. The notebook, the general JupyterLite integration in Galaxy has the ability to read data sets from Galaxy history from here. Well, in this case, the way it does it is that we just need to make sure that our GenBank variation metadata set to appropriate history IDs. Okay, so GenBank should be set to 1598 because that's the number of the data set. So 1598, 1598. Variants should be set to 1594. Okay, they are. Finally, the metadata should be 1832, 1832. And so at this point, just to make this a clean run, I'm going to go to kernel, say restart kernel, and clear outputs of all cells, and then run all cells. Now, notebook is executing through its entire content. So let's give it a few minutes. All right, so let's see what we got. This notebook was created entirely with the help of ChatGPT. So just like I have this, let's correct that error. I had ChatGPT open in one window and that notebook in the other. So let's go through the stages that were involved. One of the problems with measles, as I already mentioned, is the fact that it has a rather interesting genome organization. So you have the phosphoprotein, and within the reading frame of the phosphoprotein, you have a C protein, which has an alternative start codon and +1 phase. And you have a V protein, which shares most of its length with P, but then there is an RNA editing site where the frame is switching. So for each variant that we identify, we need to interpret its effect in two different frames. For example, here, we need to interpret it in phosphoprotein and protein C. Here, we need to interpret it in the phosphoprotein protein V. The workflow that we used, it was utilizing a tool called SnpF for doing this. But SnpF is not smart enough to recognize these issues with overlapping reading frame. So in order to actually understand the effects of variants that we have on the encoded protein, we actually need to do all of this ourselves. So this is why, again, we grab the GenBank file. So you will see that in this notebook, I actually provide the prompts that I used. But essentially, the first step here was to read the GenBank file and to convert it into a table, where for each position in the genome, it is annotated which coding frame it's in, what codon it corresponds to, and so on. So it looks like a table like this. One of the problems with ChatGPT is that it's a great tool, but you can never really trust it. So especially for complex things like this analysis, you need to have some way of checking whether the code that it's producing actually makes sense. The way we do this checking in this notebook, we actually, for each step of the analysis, we ask ChatGPT, and then we do this in Python to visualize the results. So in this step, this graph here visualizes the annotation. So if we zoom in, you will see that for all annotations, it shows you the reading frames of the proteins. And for every base, it tells you which codon position it is, what codon that is, and which is encoded amino acid. So that provides a kind of a sanity check. I literally did check this against the annotation to make sure that this parsing was correct. So you can see that this works for C protein, but let's check the V. because this is the tough one. So for V, the reading frame is, this is why it's wide here, there's nothing here, because up until this point, and this is position 2499, the V and P reading frames are identical to each other. But during the once the virus is in the cell, in some fractions of RNAs, you have an insertion of an additional G in here. So essentially what's happening is that this codon is kind of, if you will, extended, and so the V codons begin. So you can imagine that GG, and there's a additional G inserted here, and therefore everything shifts by one. So from this point, the V protein becomes different. You can see that now the third codon position of each codon in the V protein corresponds to the second codon position in P, and so on. So based on that image, I can tell that the script which we ask ChatGPT to generate is correct. It's parsing things correctly. So that provides us a kind of baseline on which we're gonna visualize our variants. So the next step is we are reading the variant table. And from that variant table, we just keep essentially the first the first 8 columns. Because again, the other columns correspond to SNP-F annotations, but we don't need them here. We're going to do this ourselves. So these are our variants. And next, again, using ChatGPT, we asked to create a Python script which would take this table, compare it with these annotations, and for each variant, this script will tell us whether this variant changes underlying amino acid encoded by the reading frame. And of course, if it's a region where, for example, where you have 2 overlapping reading frames, it would do this for both frames. It would tell what the effect of the change would be in the first frame and what the effect of the change would be in the second frame. And again, for the sanity, we are visualizing results of this, which looks like this. In this plot, we have 2 panels. The lower panel, that's the annotations, these positions of codons on the measles genome, just what we saw a few minutes ago. But on the top panel, we actually have variants. So let's zoom in. And I filtered this down, so it's not showing variants in other genes, because in this particular analysis, I'm only interested in things which are within the overlaps. So let's zoom in. For example, let's zoom in here. So these vertical lines correspond to changes. The length of this tick corresponds to the difference between minimum and maximum allele frequencies, because here we are aggregating information about each variant across all samples for a given position. So for example, in this case, if you zoom over, you will see that the minimum allele frequency here is 0.38, the maximum allele frequency is 99. It occurs in 63 samples, and actually, so in our data set, we have 225 samples. So among these 225 samples, it occurs in 63. And this is reflected by opacity of these ticks. So if the variants are really common, they occur in all samples, then these ticks will be really dark, like these, for example. So that variant occurs in 152 samples, and so on. So why I have 2 ticks here, which look identical, and I have different colors, because this is how this variant affects gene P. And this is how this variant affects gene C. So you can see that in gene P, it's red, so it's non-synonymous. Its variant changes the underlying amino acid in P, but it does not change it in C, because in C, it occurs at, let's see, so in C, it occurs at the third codon position, and in P, it occurs in the first codon position. So it's logical that it's going to affect amino acid in P and will not affect amino acid in C. So this is just like in using very simple collaboration, if you will, between ChatGPT and JupyterLite, we can generate these graphs very rapidly. But again, anything that ChatGPT gives you, you cannot trust it right away. You have some way, you have to have some way of testing it. I'm doing this by visualizing it. So next step here is we wanted to see the balance, because the trick here is what the unusual situation about this whole arrangement is that a single change have multiple effects because it affects two reading frames. So, and what reading frame is leading in the sense that it accumulates more non-synonymous substitutions, and which reading frame is lagging, less non-synonymous. So which one is more protected by purifying selection than the other? We can just do a simple tabulation. We will see that. So for C protein, we have about 50-50. So among all the changes, 14 are non-synonymous and 19 are synonymous. But in P, it's kind of the other way around and quite profoundly. So most of the changes affecting P, they result in amino acid changes. They are non-synonymous. And to make it even more explicit, we can do for every change, we can list which proteins it's affect and how it affects them. And also what were the reference codons in the reference genome and what the codons do we see as a result of the substitution. Finally, in this data set, we have results of 3 studies from Canada, from Romania, and from the United States. We also, this is why we downloaded the metadata about samples, we have collection dates. So one quick analysis that we can do is we can plot sort of from early dates. So this is what? This is 2018. And the latest samples here are 2024. There are 2 data sets where collection dates were not included. So this is unfortunately sometimes, and actually quite often, metadata doesn't have very high quality for sequencing data, but fortunately it's just the 2 samples. So you can see that there are certain patterns here. The blue here is the Canadian study. You can mouse over this to see the data, to see what it is. The middle one is the American study. And this is the most recent Romanian study. So you can see clear pattern here. They correspond to mutations, to substitutions, which are characteristic of a certain strain that we see here. But we have some interesting substitutions. For example, these changes that affect only 5 samples, 5 American samples, they're not found anywhere else. So this kind of visualization is very helpful for picking up unusual things. And again, because it's Jupyter, because it's Python code, you can perhaps view this in a different way. For example, you can ask question: Are these two linked? Are these two linked? So this one is in the position 2683. This is 2709. So in principle, it's possible if these were, for example, 300 base pair Illumina reads, we can ask the question, do we have any reads that are spanning those? Are these substitutions linked? You can do all these things fairly easily in JupyterLite. Again, the purpose of all of this exercise is to show you that you can go from raw reads to images like this just in a few hours.